So good morning. So we'll continue our sequence. So we're going to cover now new technologies for production of high value ingredients, followed by new technologies how we can use it for food products. Yeah. So some examples are from non-seafood, that means for decontamination, and we'll focus again on reduction, how we reduce the food losses or how do we transform the residues. Both the elements will go hand in hand. So just a quick recap. So we talked about biorefinery mainly related to seafoods. Yeah. So that biorefinery can be extended or can be classified based on the raw material or a feed stock, whether you consider high fat fish as a lipid rich biomass, which contains again canola, that is oil seeds or insects like that, or protein rich biomass. It can be again related to low fat fish. Fiber rich and sugar rich does not include there, but any of those feed stock could be considered as input material for biorefinery. Yeah. So when you are establishing a biorefinery, which is also part of your assignment, so this is one important thing. What are the feed stocks you are considering? Yeah. Within feed stock, what is their composition, what is their physicochemical properties, and what all you can obtain. Then comes your stabilization or post-harvest interventions or different pre-treatment. So we did discuss briefly about blanching, drying, and there are some chemical preservative in terms of salt and sugar or siling, enzymes, or using new technologies. So main focus now will be on new technologies. Yeah. And then you have a conversion technologies, that is how do you fractionate it, hydrolyze it, use enzymes or microorganisms for fermentation, or only enzyme or alone or in combination. During that you get your multiple products. All that ingredient will depend upon what sort of biomass you are obtaining. You cannot get high fiber, you cannot get sugars when you are using seafoods, because seafood does not contain much of carbohydrates and sugar containing compound. And rest of the side stream you have bioactive fibers, others, and in this process you also get a processed water, and within processed water you again can use new technologies, mainly membranes, to separate the ingredients and fractionate it further. Yeah. So this is crux of your biorefinery and how you can achieve circularity at different stages. So this is what we have so far discussed. So this is the blueprint which we did discuss in the first lecture, and then we just bringing in different processes, ingredient. So things isolating any of the processes or any of the step becomes quite difficult. So it will be some of the repetition of the examples will take place so that you can draw your own conclusion and your own links. So if you talk about all the ingredients, broadly you can classify them as proteins. You have food additives, nutraceutical, feeds, pharmaceutical, and also natural dye. Can anybody give an example of natural dye which can be used, where protein can be used as ingredient, as a natural colorant? Yesterday we did discuss. First one, phycocyanins. Red, blue, microalgae can be used directly as a food colorant. Then you have different amino acids, fatty acids, that is again to know about lipids. This lipids contains your phospholipids, your fat, oils, and fatty acids. Yeah. So this again have the similar applications depending upon the purpose. Micronutrients, vitamins, sterols, and different minerals, food ingredient, cosmetics, pharmaceutical, feed, sugars and starch, including your including all other chitin, chitosan can also be used for bioplastics, biofuel, food ingredient, pharmaceutical. So and again secondary metabolites, that is your phenolic compounds, all the phenolic compounds, and some of those astaxanthin which is coming again from seafood, and certain carotenoid compounds. They all have again the similar applications. When I say pesticide, that is basically a biopesticides. Majority of secondary metabolite can also act as a biopesticide or biofertilizer and all those applications. So, in nutshell, most of the ingredient whatever we get from different streams can have multiple applications. Yeah. So, whenever you are answering such questions, make sure that you know at least one example. Yeah. Not to shoot things in air. So, fucoidin sulfated polysaccharides, alginate from Saccharina latissima, vulvans from vulva lactum, vulva species, laminarin is almost in every seaweed, agar from Gelidium or from mainly from Gelidium, carrageenan from Chondrus crispus. Yeah. So, these are again polysaccharides. You have thickening agent as a fat replacers, as well as nutraceutical, functional food, cosmetics and pharmaceutical, nutraceutical. So, all those applications, this is all ingredients and different applications. Yeah. We can come up with the new applications, new examples, so it will become slightly messy. If you remember, we had one messy slides, big massive one, all the raw material and ingredients and how it can be used from waste as well as biomass. So, this is all ingredients and different application. Now, the question comes, why new technology? Yeah. Or what are the strategies of obtaining those ingredients? Yeah. If you remember, two approaches we did say, one was to reduction and transformation. Yeah. So, this time I start with the transformation, and then we next lecture is all related to how do we reduce it within this particular section itself. So, take an example of protein sources, because alternative protein requirement of protein is becoming quite bigger. So, whether you have existing sources or you have alternative sources, seafood, oilseed, cereals, pulses and legumes, similar concept applies. Edible insects, microalgae, seaweeds or microbial source, either yeast or so. There are only three ways of getting any ingredient. One is dry extraction, that means without using any liquid, water, or you can call it as dry process. Pre-treatment followed by dry extraction, pre-treatment followed by wet extraction. Yeah. That means giving ultrasonic treatment followed by conventional method. Giving demineralization, deproteinization can be considered as your pre-treatment followed by main extraction, depending upon what is your target compound. So, there are a number of different technologies which can be used, and after extraction you do carry out the fractionation or separation, yeah, either by membranes or by centrifugal forces to get your ingredient. So, similar figure can also be adopted for your lipids. Yeah. There how we will work on a dry fractionation, that will be again with certain limitation. If you dry it and then if you try to squeeze it, you can get oil. For example, you have oil seeds, basically it will be wet or a dry extraction. We are not adding a water, that is what we meant here by wet. And this is one of the equipment design which is microwave plus ultrasound together, which you will see in subsequent slides. This is supercritical, this is ultrasonics, this is also ultrasonic, this is also ultrasonics, and there are a number of other technologies which can be used for any of these processes. Yeah. So, here also you have high pressure processing, pulse electric field, number of different techniques can be done, and it will be difficult to cover each and every technology in this given short time. So, first and foremost, what are novel technologies? Novel means new. Oh, come on, this is going to happen again. As a new technology. So, either you have a processing application mainly for improving shelf life, cleaning, disinfection, dried food application and biofilm eradication. Why these four examples are written? One is how we can extend the shelf life of seafood or seafood related ingredients, products, or that is it, ingredients or products. Cleaning and disinfection, that means how you can clean them or how you can disinfect them, that is alternative to chlorine related processes. And dried food application, that means when you are getting an ingredient at low temperature or where you are not exceeding sublethal temperature, you need to have a technology how you can extend their shelf life. Biofilm eradication becomes an important element, because if you are using conveyor or if you are using a spray dryer, a membrane fouling, so how you can remove those biofilms. Do you know about what is a biofilm? Anybody?
Biodegradable packaging material.
Well, you can call it here, the biofilm is basically the formation of when bacteria they grow. If you spill a milk on a surface, it starts rotting, bacteria starts growing. It produces extra polymeric substances, then again more and more bacteria they grow. So they make a thick layer of microbial film, yeah. So that what it refers here, how we eradicate those films, yeah. So I know it is always a confusing word, is a bioplastic, biofilms or biodegradable plastics, yeah. So partly you are correct, but here the intention of writing biofilm is formation of film because of microorganisms or left or produce, how you remove it. Suppose you keep, if you would have seen a funnel, you keep using the same funnel all the time, there is some film development happens. Or even if you take a tea cup, you keep using the same cup, if you don't clean it, there is always a film forms. Even when you fry it all the time, you will see in the corner of the vessel there is oil, then again layer. So that is also formation of film, but that is non-microbial film. Film formed because of oils and greases, yeah. But if it is formed on food conveying system or food equipment, that creates cross contamination, yeah. So if suppose you have a vessel, or you take a fermenter and then some bit of food material is left over if you are not cleaning it. So microorganisms they grow on it, yeah. And if it is not cleaned properly, and then again second layer of food material stops on this, then again the bacteria which are already present they multiply, and then these bacteria they forms their mucus around it. Something like when you get a cough is because of bacterial infection, and the mucus what you get is an output of that bacteria, yeah. So that is formed and that has to be removed along with the microorganisms. First you have to kill a microorganisms in order to remove that mucus secretion, and to remove that, to kill that bacteria, that particular chemical should penetrate into that particular layer, yeah. So for any microorganisms to protect themselves, they make a film around themselves, and that film has to be broken, yeah. This is what it refers here, biofilm. I knew that there will be a confusion when we say biofilm, that we will always regard it as a biopolymer film, yeah. So that is what it meant here. Other one is new technologies for extraction application, that is biorefinery approach. This is what we have been covering since day one. Food waste valorization or transforming the biomass, new biomass into different ingredient like microalgae, macroalgae, we have discussed full length of biorefinery. So why we are looking for new technology? First and foremost, it ensures food safety. That's food safety is a prime motivator for any technology. Once you are safe, then only you think of the quality and quantity, yeah. There is no point in eating 10 rotten mangoes and spoil your health rather than just having a safe mango, yeah. Then improved shelf life. So use all this technology to improve shelf life by few days, few months, and reduce wastage. And when you use these technologies, majority of these technologies, they have ability to retain those nutrients and less degradation of nutrient due to different technologies. And in majority of the cases, when you consider current processing operations, they are not climate friendly or environment friendly. So we look for a technology which are more environment friendly. That means they eliminate the usage of chemicals, A. B, they eliminate or they reduce energy or water requirement. So that means all these things are helping environment some way or the other, directly or indirectly. They improve process efficiency. In certain cases, we use a technology which can improve the process efficiency rather than focusing on improving the shelf life. That means again reduction of your input cost in terms of water or energy, because these are the two important elements which are considered also for improving process efficiency, plus recovery yield. That means you can recover more. That is again related to improvement in extraction yield or purification, yeah. So application of new technologies, you would have seen or you can see number of review articles, number of published literature is there. application of new technology for A, B, C, D, E, F, G, different biomasses, different products. Every year 5 or 6 different such papers are coming in, and it may be more than that, and crux of all those papers will be is written over here. So you could put any example, novel technologies for seafood, yeah, shelf life extension, high pressure processing, pulse electric field treatment, cleaning and disinfection, that is washing of fish, yeah, dried food application if your dried fish is there, or you have fish based ingredient, biofilm eradication, biofilm also forms on the fish surfaces or conveying material, or the storage container. If you carrying a fish every time, you know, from the water, you will see slowly as a new one is perfectly fine, and if you are not cleaning regularly, it develops film, and that is sufficient enough to cross contamination. Hand is perfect example. You wash it, all things are gone, and slowly the scales develops. So that is all to do with biofilm. Then extraction application, valorization of biomass to get different ingredient from seafood side streams. Whatever waste is there, evitable or inevitable waste, how we can valorize it, or transforming the whole biomass. In case of seafood, what could be the example of biotransformation is mainly bycatches. Those fish which are caught unintentionally, and you have that biomass, how you can transform into different ingredient, yeah, and advantages remains the same. So any of the biomass can be fitted in, and you can list out all the application. So whenever you are writing such type of report, first make your own blueprint, what way you are going to frame it, and then you can just think in that particular perspective, then search a research paper, then start compiling it together, rather than other way around. First collect everything, then start building it, yeah. So that means sometimes when you are doing literature search, you just keep downloading all the papers. It's something like you keep buying the furniture and then start making the house. Do we do this way? No. We first make a house, then buy the furniture what you need. So that's that's the strategy you should be taking, what you want to write and what you want to communicate the message. Once you know what message you want to communicate, then only there will be clarity of the thought in your articles, yeah. Because science is art and science, yeah, both. Art is how you write it. Something like you are writing a letter, you want to have a essence of the message, what you want to communicate. You start with hello, how are you, all is well and everything, but your message is clear in your mind what you want to write there. But when it comes to writing a paper, you yourself are not clear what you want to write. Keep collecting it, keep collecting, writing it and saying, oh, this is a nice paper, this is a new paper, this is a nice sentence, you just keep sticking in. When you read it yourself, you will find that you are not what you are trying to say, yeah. So clarity of message is the most important. And now if you see this figure itself, if I do ask you application of high pressure processing in seafood application, yeah, then if you have printed it out blueprint, so you know where all it can be used. Accordingly, you can frame your sentences and then the way you write it, and then there is no chance of it getting rejected or having no citation. The major fault is the take home message is not clear. And if you want to see how basic and simple language articles are written, you should read the papers coming from Nature and other one, yeah. I don't think so anybody might be opening those papers thinking, oh, that's very high impact journal and high impact publication. But if you read any of the Nature paper, very language is very basic, yeah. I've seen lot of authors, what they do, they right click it and look for synonyms, which looks more complicated word, yeah. You know, just to look more fancier. That should not be the way, yeah. We try to put in more complex text, people won't understand, people won't cite it, yeah. Try to keep things simple, that's very important. Now, this one is processing of biomass for valorization. If you do remember, here I had a shell and fish. Now, exactly the same thing, yeah. Biomass and how we process it. So, I am not going to waste any further time. When it comes for extraction of ingredient or obtaining ingredient, couple of formulas are used. One is extraction yield, yeah. recovery yield, yeah, and purity, yeah. These three are commonly used. So, again when you are writing any of this, recovery yield is 70%, extraction yield is this. So, you have to understand what really you want to say about these words, because there is again no clarity of definitions related to this. If suppose you have 100 grams of raw material and it contains 30 grams of protein, yeah, protein, and this is your target to extract the protein, yeah. Now, you use whatever technique you used and you get your crude extract, yeah. Suppose crude extract after drying you get around 10 grams, yeah, and your residue is 90 gram, which is not possible, some will be lost. Take it as residue is 80 grams, yeah. So, once you have 10 grams, what you do is either you do the precipitation, yeah. If you precipitate it and then after just take it as 100 grams of fish, yeah, what you would have done experiment, crude extract, that is alkaline extract, you got it as 10 grams, which is written on dry basis, or you can in a you can convert into liquid, you will get it in liquid form. After precipitation, suppose you get 4 grams, yeah, and this dried residue is 80 grams, yeah, and in this 4 grams the protein content is around 70%, yeah. So, the protein purity here is directly estimated as 70% from calculation, yeah. So, what will be the extraction yield, yeah. Now, extraction yield will be protein yield, either this 4 upon your starting material, that is 100, yeah, that is 4%, or crude extract yield will be 4, if you take it against the crude extract 4 upon 10, it becomes 40%, yeah, but that will be again recovery of protein, that is 40% in your this extract itself, right, yeah. If original protein is 30 grams, so maximum protein what you can recover from here is 30 grams, yeah, and here you have 4 grams, and 70% is the purity. So, 4 into 70 is how much? 2.8 grams, yeah. So, you recover 2.8 grams protein. So, 2.8 grams and original was supposed to be 30 because it is 100 grams, yeah, upon 30 into 100. So, this is your recovery yield, yeah, okay. And there is, this is one system, right, and then this is your suppose a control process. Now, you use ultrasound or enzyme or something, yeah, there everything remains the same way, whereas here you got 3 grams, yeah, and your protein percentage in this is 99%, yeah. So, which one is better process? Even though here crude protein is less, yeah, but purity is high, yeah, and 99 into 3, same thing here, yeah, 99 into 3 upon 100 divided by 30. So, that is why sometimes even the crude extract yield is less, but recovery of protein is more. So, it cannot be that extraction yield is high, recovery yield is high, purity is high, possible, yeah. It is possible the purity is low, recovery yield is high, extraction yield is low, yeah. So, when you are writing any of those things in any of your manuscript, better to state the formula, even though it looks like this upon this into 100, that is it, that is fine, yeah. And then non-recoverable protein, again you multiply this into this, you get your non-recoverable protein. So, that is the way you can make your mass balance completed, yeah. Sometime it happens you get your crude extract, yeah, or recoverable extract, this plus this is more than your starting material. Suppose you started with 100 grams, I am just stopping it here, crude extract you got 30 grams and residue you got 75 grams. Is it possible? One possibility is wrong weighing balance. Yeah. Second possibility is 100 gram was having 10% moisture and this was having 5% moisture. Yeah. Or this was having high moisture. Yeah. Another thing is it also depends upon what solvent you are using. If you are using 2 molar NaOH, that means you are adding salt also to it. Yeah. So, that dried ingredient will be added here or here. Yeah. So, not to think that you did not weigh it properly, just weigh it what all is going in and whatever is coming out. In some cases when you use enzyme for protein extraction, there your protein yield is purification is high. But remember, when you are adding an enzyme, you are adding 100% protein to the solution, to the system. Yeah. So, all those things has to be considered, but you can compare like with like. Control enzyme plus ultrasound control enzyme with microwave, then it is fine. Yeah. I am 2 meter 1.8 meter tall, another person is 3 meter tall, but he is standing on the stool, or other person is standing on the table. So, there has to be level playing field. Yeah. That is why every height is measured based on mean sea level. Yeah. It is not what is high, what is low. So, you have to make benchmark as straight. Even if you are using HPLC, GCMS or anything, you have to make a baseline straight. Yeah. So, if you have any question related to that, keep it ready. I am happy to answer that. If you want to see mass balance, I can show it to you at some point. Yeah. Now, during the process we did talk about clean or green processing technologies. Yeah. So, what do we or how do we define clean or green processing technology? Clean in the sense is not clean vessel. Clean means which does not have a negative impact on environment, or whatever end product you are getting or processes you are deploying do not have negative impact on environment. Yeah. That is what it meant by clean and green. So, first and foremost principle number 1 is raw biomass. So, in terms of circular economy or blue circular economy or bio circular economy, raw material should be from renewable sources. Yeah. So, that it is not depleting resources. 1. Second principle is associated with the solvent. That means the solvent what you are using is a clean or green solvent, or GRAS. What is GRAS?
Generally recognized as safe.
Can you name one? Water, salt. Yeah. So, that is even ethanol generally recognized as safe. That means they are safe chemical, but that safety does not mean they will be safe. So, that is one of those examples. So, the most important and easiest solvent is the water. Yeah. Then comes your alcohol or alkali and all those things, they do come in. And principle, that does not mean you replace that particular solvent completely. There is another way that you can reduce the usage of that solvent. Yeah. So, still the challenges are how you can find natural alkali. You get lot of natural acids, but finding natural alkali is difficult. Can you think of any alkaline substance available naturally, any fruit or vegetable which is having pH more than 8? So, that is why one of the famous doctors said, if you make system alkaline, there won't be any cancer. Majority of the system is acidic. You take a strawberry juice, add alkali to it, red color is gone. Yeah. So, that is why its natural alkali is one way how you can get alternative to alkalis. You need to find alternative to alkali. Second one, what is alternative to hexane? Diethyl ether, petroleum ether, those are all again chemical. So, hexane is extensively used for extraction of lipids. One is of supercritical CO2. Is there any other solvent which can be used as an alternative to hexane? One few new chemical has come up, that is TMO, yeah, or THF, yeah, which is said or which has been claimed to be a better than hexane, having less of cytotoxicity or so, yeah. So, we have one of the PhD who is working on a specific chemical which is considered as an alternate. I will get you the exact scientific name of this, which is alternative to hexane and having a better application, because hexane problem is it is highly flammable, yeah. At the same time it leaves the residue. So, we need to find an alternative to hexane or non-polar solvent. So, this is to do with the solvents, and there also your deputatic solvent and all those things comes into play. Third one is related to energy. That means any technology or any processes we use should have lesser amount of energy requirement. That means you are saving energy. When you are saving energy, you are benefiting environment. That is another one. What could be the fourth principle? The process, yeah. That means whatever process you are suggesting, that particular process should not have a negative environmental impact. For instance, you do not want to have a technology prepared or developed in such a way that is heavy on resources. You have renewable biomass, good, you have water as a solvent, it takes less energy, but to make that particular process you need certain ingredient from moon or something else, yeah. So, that is also depends upon what type of processes we are making, and these processes should not be heavy on, for example, on infrastructure, building a new structure, or the structure can only work underground. So, you have some other issues, or it only can work inside the sea, then disturbing other processes. So, this is to do with process, or should also be you should be able to replicate that, yeah, at a commercial scale, or the scale where you can really talk about environmental benefit. Fifth principle is related to target ingredient or extract. Even though you have everything perfect, the target ingredient you extracted is good, but you cannot have any application of that because it does not have those properties, yeah, or they have a high level of cytotoxicity, or their biological activity is gone, yeah. So, that is another principle. And the last one is related to waste and byproduct. First and foremost, it should not develop any waste. If does develop or forms and waste, that waste should be environment friendly or can be used for composting or for fertilizer purposes, yeah. So, this is what a principle for clean and green processing technologies are, yeah. Is it clear? No? Good. Now, first one, as I mentioned about the diagram, dry extraction or wet extraction and pretreatment. So, what is basically a pretreatment? That is any unit operation given to a biomass in order to improve extraction efficiency or to stabilize it. So, take this particular biomass that was the easiest one to draw, that is why you see all the bamboos here, yeah. So, this is the biomass and your all the ingredients are within that particular biomass. You have a protein, you have fiber, and you have different stuff within enclosed into that particular biomass. So, what sort of pretreatment we can do, or what sort of pretreatment you are aware of? You would have studied every of these ones, yeah. You divide it into a chemical, liquids, organic solvent, alkaline, acids, biological, that is microbes or enzymes, yeah. Your physicochemical, that is hydrothermal, heat and moisture together, steam. FX, this is actually accelerated solvent and fluid extraction, these are physicochemical. That means when you are taking mechanical and chemical things together, or milling, grinding, chipping, or slicing, maceration, all those things can be classified as a pretreatment, yeah. why we do this pre-treatment? To disrupt the cells. Yeah? When cell is disintegrated and then you add your solvent, and when you add a solvent, solvent will dissolve your target compound. Suppose you want to extract oil from the protein plus oil suspension. The moment you add hexane, only the lipid will be dissolved in hexane, yeah, not the protein. But if you add a water, only protein will be dissolved in a water if it is soluble in water, yeah, and then subsequent processing you get your product. So this is your extraction system, this is your pre-treatment, and whatever pre-treatment we apply, that affect both upstream and downstream processing. Downstream processing this one, upstream will be on this way, yeah. That means whatever residues you are getting, that disturbs that. So, so now if you see all these methods again listed out in clear sense. So what will be the best pre-treatment or what will be new pre-treatment? Chemicals, sorry, physical one, reducing size. If you grind it, take example of shells. If you make finer particles, it will have better extractability. Size reduction, that means per unit surface area is increased. Densification, grinding, milling or membranes, that is you sieve it out. Chaffy material, if you are doing grain protein extraction, you remove the hull which does not contain a protein, then it will have a better yield for proteins, yeah, that all physical. Chemical, we just discuss all those chemicals. Biological, microorganisms or enzyme, yeah. Now within this here we have clean and green chemicals, that is your water, your ethanol, yeah. What else? What other solvent can be considered as clean and green chemicals, yeah. You have deputatic solvent, you have a salt solution, or you have use of organic acids like lactic acid, yeah, citric acid, acetic acid, all those things can be considered as cleaner one because they are all biological origin. Now what is the best part is also to have a combination of these, yeah. That means heat and water together, steam, combination of physical and chemical approaches. Sometimes you have to combine two to become different types of pre-treatment, yeah. Any questions so far? No, okay. Now this is quite a complicated one, yeah. So you have to pay special attention to this, yeah. So this was one, biomass. I already mentioned every system does not need a pre-treatment, yeah. In some cases you have to have a pre-treatment, some cases you do not have to do it. Now this is how we develop a extraction process. Novel techniques, extrusion, compression, microwave, ultrasound, supercritical, enzyme, yeah. Although these all are not new, milling is not that new, but again how you mill it, that will be classified as a new. Conventional, grinding, milling, alkali, ethanol or formaldehyde, which we mentioned during alginate extraction process. So pre-treatment whether with a new technique or with conventional technique, yeah. Then comes, you can just link anything here. Again novel extraction, as you can see some of the techniques mentioned here are also mentioned here, yeah. Biomass pre-treated with new technique with novel extraction, yeah. Biomass pre-treatment with conventional technique and then you have new extraction, yeah. Biomass no pre-treatment followed by straight away for extraction, yeah. Or you have combination of novel extraction, that is ultrasound and microwave together, enzyme and ultrasound, ultrasound to disrupt the cell and then to add enzyme, or you add an enzyme first and then to use a microwave to inactivate that enzyme and carry out the downstream processing, yeah. Use green solvent or non-green solvent, yeah. So as I said, one of the principle for clean and green extraction principles, if you are using acid, alkali or different chemicals and if you give a pre-treatment with ultrasonic, your usage of solvent reduces, yeah. instead of using 2 molar, you may get away with 0.5 molar or something. So, reduction on usage of solvent. So, you can have this, you can have this, yeah. You can make a number of different combination. The purpose is which works for what biomass, yeah. So, that is all to do. Every biomass does not need a pre-treatment, that is one message. Second one, pre-treatment can be with a conventional, with a new technique. Extraction can be new pre-treatment followed by conventional extraction. You will see some of the example in coming slides, or pre-treatment with a new technique, new extraction and new solvent, yeah. So, any aspect of this will be considered as novel extraction technique, yeah. So, novel means new, is not that it emerged yesterday, yeah. So, it's novel means it's still, we keep calling it novel till the time it is having application at commercial scale, yeah. It's very similar to pathogens, we call it as emerging pathogens. Can you name any emerging pathogen? Say for example, E. coli, yeah. When it was discovered 200 years ago, but still we call it emerging pathogen, yeah, because it keeps emerging in a new form, something coronavirus, it's not gone, it keeps emerging in a different forms, yeah. So, same way it will be called new technologies, even though some of those ultrasonic is been used in 1950s, yeah. So, it will be called till the time we have final application, and you will not be able to find or clear definition what is the new technology, yeah. Okay. So, that is one. Now, conversion system, non-thermal processing, novel pre-treatment and novel extraction. Now, the word non-thermal comes in here. Certain processing technologies which are considered as non-thermal can also be used for extraction purposes. That is the place where we have significant overlap. Purpose, enhanced recovery, energy efficient, that means retains the quality of that ingredient, add value to the side stream and helps in waste utilization, yeah. So, wherever you see in my slide, wherever it comes to technology, you will see exactly the same pattern and only some of these points are different, yeah. But crux of the matter is these technology remains the same. This is high pressure, ultrasonics, plasma what you have seen here, cavitation, this is cavitation, LED light, ozone system, yeah. So, can you name or do you know how many novel technologies are there at the moment? What you are aware of? Anybody?
Sir, pulse electric field.
One second, yeah. PEF, yeah.
Cold plasma.
Cold plasma.
HPP, high pressure processing.
Yeah.
Ultrasound radiation.
Yeah. Ultrasound.
Microwave.
Okay. Microwave.
Ozone treatment, ozone treated waters.
Ozone, yeah.
Irradiations.
Irradiation,
techniques.
okay. And I will put it as a gamma, yeah.
Gamma radiation.
Yeah.
Some novel drying techniques like infrared drying.
Okay, infrared is another one, yeah. Infrared is drying as well as processing also, okay. Infrared IR, yeah. Anybody want to add anything else?
So, ohmic heating.
Okay.
Inductive heating.
Ohmic heating, inductive, yeah.
Shock wave technology.
Shock wave. Any other addition?
Supercritical fluorescence.
Okay. SFE, yeah.
dense phase carbon dioxide.
That is supercritical CO2, yeah, okay. Dense phase is supercritical, yeah. Then you have subcritical water extraction also, right. Any other? I am also thinking. Any other?
UV radiation.
Okay, yeah, UV light, yeah.
Pulsed light.
Pulse light, yeah.
Sir, all those assisted extraction, UV assisted.
Yeah, that's assisted will be like, I am just talking about the main one, yeah.
Okay.
Like we have standard units and they have derived units. So, that all assisted you add it, all will come in there, yeah. So, all these ones, whatever is listed here, they all are non-thermal, yeah. All whatever microwave, IR, ohmic, inductive, induction, and all those are novel thermal, yeah. Novel non-thermal, novel thermal technique. The only difference between both the technique is novel non-thermal, where you achieve microbial inactivation at sublethal temperature, whereas novel thermal, temperature is the prime motivator, but distribution of energy is efficient. Microwave distribute heats uniformly, whereas thermal processing, it is always conduction plays a major role, yeah. That means from outside to inside, whereas microwave, ohmic and all, they all have a uniform heat distribution. So, they are more efficient. So, novel thermal, novel non-thermal, they are two different elements of it, yeah. Now, if you see, majority of these technologies are only few of those can be used for dry material, and majority of them are useful for liquid food application. Supercritical fluid extraction of CO2 or dense phase, that means you are changing the density of CO2 from gas to liquid and carrying out your extraction, yeah. So, other ones are also there, like natural antimicrobial agents, bacteriocins, you have chlorine dioxide, and all those are also classified within that. So, there are almost 15 to 20 which have shown different application at different products, yeah. So, we will only cover few of these techniques and rest all are there anyway, because that could be a full module on its own when it comes to technologies, yeah. So, now we start with membranes, yeah. Now, membrane, that means whatever solid products are, will have subsequent example for extraction, another one. The important aspect what I was trying to cover here, we came across a lot to replace ethanol with membranes, yeah. What is basically a membrane? Membrane is a simple sieving system, yeah. Simple sieving is there to remove solid with liquid, yeah. So, is a layer of material which serves as selective barrier between two phases and is impermeable to specific particle, molecule or substances when exposed to action or driving force. Take an example of milk. You curdle a milk by adding a lime to it, yeah. Then you have muslin cloth, you put into it and then you driving force is a force. So, all the way goes out and you are left over with cheese or paneer, yeah. So, that is what a membrane is, yeah. Simple membrane is your cheesecloth membrane. Same thing of metallic mesh, that drives based on size. Now, once you get the way, but still it is having a sugars in it or some proteins in it, how you separate that? If you put another membrane 3 times segregated, still you will recover some more, right. So, that means you need more and more force to separate things, but at the same time you do not want to put that much of force that membrane collapses, yeah. So, if you will see here, simple process, this is your membrane and you pass through a liquid, one is retained on the top and one passes through, yeah. What passes through is permeate, what is retained is retentate, yeah. So, some compounds allowed to pass by a membrane into permeate, whereas other are retained and accumulated in retentate stream, that is retentate stream. So, this is what a membrane processing is, but is this as easy as what is seen here? Will complicate it now. So, different types of membrane depending upon the driving force. One is your microfiltration, which removes the particle based on at a micron level, ultrafiltration at a molecular level, reverse osmosis, that means everything is gone, and then your nanofiltration. Yeah. So, as you can see, different particles based on the particle size, these can be separated. We all know about RO water, that means everything is gone, reverse osmosis. Yeah. Now, what happens? You have membrane and you put a pressure and it removes the substances out of it. We should not relate both microfiltration and ultrafiltration as the same one, because in ultrafiltration we are talking about molecular weight level, that means proteins, sugars, and all those stuff has to be separated through ultrafiltration, which is based on molecular weight. Yeah. So, it is not that you have whey and you put it straight to ultrafiltration, it will clog. So, first you have to do a membrane processing using microfiltration to remove the larger particle, like what you do in PTFE filter for HPLC. What is the size of that membrane? 0.25 micron or 0.45 micron or 25 micron or 45 micron. Yeah. This is easy to remove all the particles. And remember, membranes are also used to remove the microorganisms, because these are all microns. Yeah. E. coli size is what? 0.1 micron, 0.2 micron. That is why you need to see microscope or scanning electron microscopy. So, these also removes the microorganism. That is, membrane filtration is also used for processing or preservation of food purposes. Yeah. Then you have based on vapor pressure or concentration differences, that is dialysis. Dialysis is very similar like kidney. If you have a salt and a protein, you put large amount of water, salt will automatically move from higher concentration to lower concentration and protein is separated. Then in some cases you need to put electric current, temperature gradient or heat plus osmosis. Yeah. So, I am going to quickly cover through the membrane, not going to the details of all the calculation and other things. So, microfiltration 0.02 to 10 micron. Yeah. Driving force is a pressure. Yeah. Mechanism is like a simple sieving. So, most of the microalgal cells can be removed by using microfiltration. Ultrafiltration, molecular weight cutoff. So, molecular weight is in, what is the unit of molecular weight? Yeah, Dalton is correct. What is, how much is 1 Dalton? Nowadays, we'll play musical chair. Whosoever is having a mic will have to answer it. Yeah. So, how much is 1 Dalton? If I say molecular weight of glucose, oh sorry, C6H12O6. What is the molecular weight of glucose? 180. How do we get it? 12 into 6, 12 into 1, 6 into 16. Yeah. So, 180. So, 180, this is 180 grams per mole. Yeah. So, what will be in Dalton? 1 Dalton is 1 gram per mole, am I correct? Yeah. So, when we say Dalton, it is gram per mole. So, I hope you all remember what is SI unit. SI stands for Systeme International. So, what is SI unit of light? Candela, CD, yeah, that's fine. Okay, anyway. So, this is what is your molecular weight in terms of Dalton. Yeah. So, it is only 180 Dalton for glucose. And how we determine the molecular weight of protein? Can we count all the carbons? Can we count all the hydrogen and oxygen? Difficult. Yeah. So, you have to use a certain size exclusion chromatography to determine the molecular weight. And how that operates? You put a known molecular weight compounds into it, you get your standard graph and then you determine the molecular weight. Otherwise, you have to determine all the amino acids and then you have to determine their all number of carbons, hydrogen, and then you have to calculate it, and that comes in terms of kilodaltons. Yeah. So, large molecular weight, smaller molecules. So, there you have to use ultrafiltration. That is again micron is one thing and actual is molecular weight cutoff. Nanofiltration, reverse osmosis, all are gradients related to pressure. Yeah. So, it does not mean if you are using an ultrafiltration membrane, it will separate on its own, or you exert huge amount of pressure. If you exert huge amount of pressure, still other things will also pass through. Yeah. Sieving, solution diffusion, adsorption, capillary flow, solution diffusion, and both counter ion and this. Sometimes you have to use ionic membranes, so that anything whatever is having a charged particle is retained, rest all passes through. That is mainly used when you are targeting two compounds which are having a similar molecular weight range. Yeah. And then electrodialysis and vapor pressure and pour evaporation. So, these are all membrane systems and how you develop into the process. Yeah. Now, types of membrane: polysulfones, cellulose acetate, polyether sulfone, polyethylene polypropylene, PTFE, which is extensively used in HPLC and different gas chromatographic analysis, PVDC, PVC, carbon filter. So, these all are different types of membranes which are used. So, can you name what will be the pore size of this membrane? This is not the pore, membranes is on top of it. Yeah. So, this is the configuration how membrane is pretty much used. Inorganic, that is glass membrane, metallic membrane, and ceramic. So, they do have very good thermal stability and withstand chemicals, because if you are using for example cellulose acetate membrane and you are trying to remove acidic material, hydrochloric acid from protein, so that might impact the performance of that membrane. Yeah. So, selection of membrane largely depends upon your type of your raw material, what you are trying to separate and what you are trying to separate. Yeah. So, there are two things, one is what is the material and other one is what is your biomass. You cannot use a cheesecloth to filter acid with other substances. Slowly repeatedly using hydrochloric acid will damage those fibers and the gaps may increase in size. So, that is why selection of membrane having certain criteria which we will cover in next few slides. So, configuration. This is a membrane configuration, that is dead end flow. This is the membrane what you saw in previous one, something like this. This is end. So, all the liquid goes in here, whatever is not required comes out. Yeah. Whether it is having a spinning configuration or a pressure configuration. Cross flow, that means a liquid comes through here, your permeate comes over here and retentate pass through. Yeah. Transverse flow, membrane is in this form and this is the way it works. That means permeate comes out of this, retentate comes out of this. So, this could be configured something like this. That means your liquid is coming in here, yeah, in this membrane, and only the permeate goes through this, and there is an arrangement done. So, this is all this is all what way the membranes can be configured. Yeah. And majority membrane systems are people, they build their own membrane system specific to the processes, or membrane systems are bought and different membranes are purchased to do that filtration system. Now, here again some of the calculations are there, what is the feed, what is the performance of membrane and how much permeate is coming in and what is expected. Dead end flow or cross flow, the simple configurations here for membranes. So, again hollow fiber, microfiltration, ultrafiltration, and these are the some of the fibers which are used for membrane. Yeah. Do you think membrane is the best way of filtering substances? Looking at these slides, yes it is. Yeah. But how many times you can use a specific membrane? because certain membranes should be always in a solution. It is not like a tea filter, you clean it and tap it and keep it and use it again. Some of the membranes has to be always in a specific solution, because if they dry off they won't work, yeah, and their configuration and chemical composition might change. Spiral wound, or simplest one is a flat membrane. We use a lot of flat membrane systems, yeah, and other geometry, hollow fiber membranes, tubular element, spiral element, plate and frame membranes, yeah. So, all these are, or membrane is not a single stuffed membrane, processing itself is a full length technology, yeah. So, that is the way we can configure a membrane depending upon the application, and lot of suppliers are there, lot of manufacturers are there who make membrane systems. As you can see, reverse osmosis RO water is used almost in every food processing plant, yeah. So, membrane system is not expensive. Processes can be expensive, but they are much cleaner processes. Majority of whey protein isolates or whey protein concentrates are made based on membranes. The problem comes today, if you are using whey, tomorrow you want to use fish, day after tomorrow you want to use something else, yeah. So, for investigation purposes, for research purposes, membranes are tested and then they are expanded, yeah. And there are a lot of large scale operations are done even to remove lactose from whey isolates. So, that means sugars and other elements can be removed. So, what should be the basis of membrane selection? One, fluid. What are the properties of fluid? That is pH, temperature, etc., dissolved solid content, and then you have your molecular weight, physicochemical properties and their targeted species, nature and loading of any suspended material, and deciding membrane type, membrane material, molecular weight cutoff, and configuration or module, yeah. It may happen you use a membrane system to segregate the stuff between 100 to 150 kilodalton, and then when you check its molecular weight, it is completely different. Is it possible? If you use size exclusion chromatography to measure a molecular weight, what is the unit of molecular weight you get there? Kilodalton, and that is basically a distribution what you get, something like this, yeah. The molecular weight which is recorded is mean molecular weight, yeah. So, there is always a mean molecular weight, and then you have mu plus minus standard deviation, yeah. So, you get a distribution and what is the mean molecular weight. So, membrane will not do it that way, yeah. So, that is why there could be a difference between physical membrane system and chemical way of measuring molecular weight, because in chemistry you get all molecular weight distribution, bimodal, unimodal are drifted towards, whereas in membrane you get completely different. So, it should be within that particular range. So, these are the way how you can select membranes. Now, we talk about the problems, yeah. First and foremost is fouling. That means, you have seen when you use a sieve, some of the stuff is trapped between the mesh. Suppose it is of 2 micron, so particle is exactly 2 micron, it will clog, yeah. So, that is again formation of gels, pore blocking, caking, that is cake layer is formed, or because of pressure some of the membrane pores are wide open and stuff is stuck. Organic matter colloidal suspension, that is really soluble salts or biological growth. Here is also the thing of biofilm. If the same membrane is used again and again, some bit of milk protein is left over there, it causes membrane fouling, microorganisms they can grow, then you have to clean the membranes. Cleaning of membranes are also done by using chlorinated compounds or certain chemicals. Even plasma is also used for cleaning of membrane as an alternative to usage of chlorine, because if you use a chlorine for cleaning of membranes, the chlorine residues may be left in a membrane and that will pass it on to the product. This is was some way or the other it shows how the fouling layer can work, yeah, and then how it is clogged between the membranes, cake formation, or because of air bubbles, yeah, and then how it can impact membrane. So, membrane geometry also plays a vital role in this. Spiral membranes, ceramic membranes, they are much better, quite expensive, as well as membrane fouling. So, here also again new technologies are used to improve the membrane filtration processes by using ultrasonics or techniques to clean those membranes, yeah, and how often you need to replace the membrane. It is not that membrane once installed can be used forever. They also have a specific life of usage. Just whenever you talk about membrane, think about your tea strainer, yeah, how long you can use it. So, that is the issues with membrane fouling. Any questions so far? Yeah? No? How many of you have used membrane system? All those who have drank tea, they would have used it at home anyway, yeah. So, membrane is not always ultrafiltration or nanofiltration, yeah. So, this is quite efficient. Microsystem is good enough to remove those, and it can replace the centrifugation, because when you use centrifugation, then you pour the suspension, then again you have some of those middle layer which you can have some certain particles, yeah. Some cases we do go through centrifugal force before going for ultrafiltration. That is also done, so that we do not have to use microfiltration followed by ultrafiltration, and same system can operate all different types of membrane, but you have to change only the pressure gradient and crux of it, yeah. Now, this is the last slide for this lecture. One is membrane separation for application to drinking water treatment, process wastewater, all those things. Molecular weight, relative size and separation processes, yeah. If a large molecular weight, that is again your proteins or so, then you can use ultrafiltration, microfiltration, or simple filtration media. I mean, sand is the best filter, yeah. If you see the domestic water filter columns, what they have? They have activated carbon to get rid of the smell, you have sand in it and enclosed into a particular sieves. Water comes in, sand is there, you have activated carbon, and it removes most of the impurities and bacteria and other element, yeah. Or you have again ozone system along with it to kill those microorganisms. So, bacteria, algae, sand. So, this is all those microalgae is sufficient enough to use a normal filtration system or microfiltration. You do not have to go for ultrafiltration or reverse osmosis. But if you are using, if you are growing microalgae and you want to segregate the protein simultaneously, if you remember that particular diagram, that means you do a pre-treatment to the biomass and then you use directly ultrafiltration after microfiltration, so that you can separate the proteins or nutrients directly out of it. So, based on the molecular weight sizes in terms of kilodalton, what type of stuff are required. So, if you have to remove organics, soluble solids and even salt and ions, you need RO or nanofiltration, which is extensively used in drinking water treatment, yeah. Any questions so far? It was pretty much straightforward, yeah. So, what we covered is how we can separate, or what is the principle of clean and green extraction technologies, why do we need to use it, and the simple technique of removing the solid from liquid side stream, yeah. So, when we come back, we'll go through the processes for ingredient side of it, solid ingredient side of it. Yeah. So, we'll come back in Continue with the novel ingredients and technologies for food application. And the previous one what we went through was the membrane processing, process for clean and green extraction technologies, and this will be in continuation for next 3 or 4 lectures are going to be technologies related to ingredients, food, feed and biopolymers. So, there will be some bit of because technologies cannot be used in isolation. So, you will see some of the examples and you have to draw out your conclusions, and any questions are there you will be asking. As we said, tomorrow morning we will start like happens in parliament, question and answer session. So, each one of you have to come up with a question. Yeah. Okay. So, now we will continue with novel ingredients and technologies for food application. So, one of the most important ingredient itself is a protein. Yeah. Protein directly as a food application or protein as an ingredient, protein as a food product. So, how do we extract protein? It's pretty much a straightforward process. Yeah. And the flow diagram what you see on your on my right side is whatever is the source of the protein, pre-treatment, grinding, milling or defatting, removing lipids, alkaline extraction, centrifugation, you get alkali extract. Here instead of centrifugation you can have a membrane to remove the alkali versus residue. Then use a conventional method of isoelectric separation or precipitation. You centrifuge it, again you can use a membrane here to get your protein pellet. This is pretty much standard process everywhere used for protein extraction, whether it is grain, oil seed cake, fish, what you have already done, or whelk or shells. Any of the biomass, this is pretty much the standard process. We have already discussed the concentration of alkali, ratio weight by volume, pH 8 to 12 or whatever depending upon the best solubility profile of a protein, targeted protein, time and temperature. So, how do we decide what is the best pH for protein extraction? One way of doing that experiment is, if suppose your target is fish protein from salmon or any source or from chickpea or any of the grain crop, buy that commercial protein and make different pH alkaline solution and to see where is the maximum solubility. Yeah. A, that will tell you what should be the pH. B option, what should be the ratio. Yeah. If suppose your ingredient contains 20% protein, that means in 100 grams you are using a biomass, that is 20%, and what is the solubility of that protein in alkaline solution? That is indirect way of measuring what should be the ratio. Yeah. Other one is from the literature. Yeah. But literature is better to confirm yourself. While doing so, you will come up with your better idea. To start something, to achieve something, start something. Yeah. Otherwise, you will be keep reading, keep doing it and keep making permutation and combination, and when you go and doing experiment, you will be starting at completely different pitch. Yeah. So, that is all to do with selection of conditions. Same thing applies for, yeah, Mike. Mike.
Sir, could you just elaborate thing on the ratio part? Means
okay. Now, when it comes to ratio, I'll just go on. Okay. When it comes to ratio, yeah. Now, if suppose I will put it in this way, because I did discuss this before. If you want to dissolve 100 grams of sugar in water, yeah. So, how much of the ratio of the water you should take? Equal amount of water, double of it, 10 times or 50 times. More and more water you add, faster will be the solubilization. Yeah. So, that is one thing is solubility of that particular ingredient in that particular solvent. I will make it more complicated. If you have 10 grams approximately sugar in sand, and your purpose is to remove that sugar from sand. Yeah. So, how much water you should use depends upon 3 factors. Number 1 is the solubility A, B, how much water you take it out, how much ever water you are using it, you have to remove that much amount of water. So, there will be energy requirement. More and more ratio you put in, better will be the extractability, but you have to compromise between its removal efficacy. Once you take a solvent, then you have to remove the solvent. So, that means you are inputting more and more water. 3 factors, remember, solubility and amount of energy it will require to add water and the cost of the solvent as well. So, those 3 things becomes an important part when we use the ratio. Yeah. So, it all depends upon what is the solubility of your targeted compound in that particular solvent. That is also the decision to see whether you can have a mixture of solvent to get better solubility. If you see in the case of bioactives, mainly water is used or 80% methanol is used, yeah, because methanol can bring in polar and non-polar phenolics as well. Yeah. So, that is all depend upon your target compound. Yeah. Did I answer your question? Yeah. Sorry. So, that is one. Now, how we can use a new technology? Technology either in protein solubilization and separation by membrane filtration. Agree? Over here. Protein solubilization, membrane, and then straight away spray drying without going for isoelectric precipitation. In case if you are using water for separation of protein, if your biomass, for example, duckweed is having large amount of rubisco protein which is water soluble, in that case you may need not to go for precipitation of protein. You solubilize it, use a membrane to remove the sugar fractions, and the left of the biomass goes straight away for spray drying and you get your protein. Yeah. Or else use an alternative technology to extract. That means you use ultrasonic to reduce this time to couple of minutes, or microwave to reduce it to couple of minutes, and then reduce the usage of solvent and then carry out with the conventional method. Yeah. This will be quite clear when we see this particular system. This is hydrodynamic cavitation, is another form of ultrasonic system where we use the dynamics of water to create cavitating bubbles. Cavitating bubble, that means cavitation is formed because of dynamics of water. This was the system designed with the patent of this particular cavitator with the University of Turin in Italy and with our industry manufacturer Cavimax. So, they designed it specifically for our purpose. If you will see, these are A tank and B tank, there are 2 tanks having a stirrer in between where you put your biomass and a water or your solvent. Mainly we use water. This is just a normal pump which is pumping it through a cavitator here. This is only the pressure meter and all those things, and inside the cavitator there is an inlet and there is an outlet, and there is a rotor and stator arrangement, as you can see here, rotor and stator arrangement. Because of this, it creates lot of cavitating bubbles which either implode or explode. Explode, that means it blast out because of internal pressure, or collapses because of the gradient between external and internal pressure, and during that process it produces lot of cavities. And the way it works is either it is a rotor and stator, this is static, this is moving, or this is moving, this is static, or both are moving in different directions. The number of different configurations are there. Other type of cavitator is venturi type, that means liquid is flowing through and you have tiny constriction over here. Because of that, it produces the cavities. Take it in a way, if you turn on a tap, water flows in a laminar flow. If you increase the intensity, the diameter remains the same, what will happen? A lot of bubbles are formed. Yeah? And if that is happening at very large amount of pressure, huge amount of cavities are formed. Yeah? So, that can also cause a damage to the product. Yeah? So, in a similar principle comes with hydrodynamic cavitator. So, if you will see over here, any of the given biomass which goes into this, it creates into a slurry. Yeah? That means it disintegrates the biomass into finer particle. Yeah? similar type of stuff can also be done with a colloidal mill. If you have wheat grain, you soak it in water, or you would have seen idli or dosa batter, yeah, it's a grain, a stone grinder, it makes into the paste. Then you want to extract something, it becomes easier. Similar thing can be done with using hydrodynamic cavitator, yeah. Now if you will see this one, HDC plus isoelectric separation or precipitation, whatever you want to call it. Technically it should be precipitation, sometimes we put isoelectric separation. Protein content and recovery. 1 pass, 2 pass means nothing. There is no unit called 1 pass, 2 pass, yeah. That depends upon the flow rate and other condition. Just due to the commercial sensitivity of the information, it is just written as 1 pass or 2 pass, yeah. And then with this itself we can get that much amount of recovery and the protein content coming out of the biomass, yeah. So here we just use this one as pre-treatment for separation, and if you want to use alkali or any other acid to it, in this machine it can be used because all those seals and gaskets are made up of Teflon. So they are resistant to any of those corrosive chemicals, yeah. And during the cavitation process, since the water is having huge amount of dynamics, it produces heat as what happens in most of the ultrasonic cavitation, yeah. So this is a cavitation based on dynamics of water, yeah. So and then this is what different types of rotor and stator it works, and all these things causes bubble formation, and it will be much more clearer when we go into the mechanism of ultrasonic cavitation. So now coming back to your sort of biorefinery stuff, whether it's microalgae, Cholera vulgaris, nanoscleropsis, you dry it, you get dried powder. Either you use a fresh paste of microalgae when you remove the water through santification or through membrane concentration, or dry powder, you treat it with high pressure to break the structure, to dislodge the structure, and followed by ultrasonic system or with a microwave, or after that this can be considered as a pre-treatment and extraction to get your lipids. Once you get your lipid, you can go for further enzymatic extraction or ultrasonics to separate protein, or high pressure followed by this, yeah. So all those processes can be established, and when you are trying to do that valorization part, whatever biomass is, even if you are just going with high pressure treatment and then to see their digestibility, because these technologies not only improve the extractability, but also can improve the digestibility and functionality of those biomasses, because microalgae on its own itself is protein, yeah. If you take for example spirulina, which can contain up to 60 to 70% protein, and protein concentrates are already 60 to 70%. Cholera vulgaris, depending upon again the strain, it can have between 25 to 45% protein, yeah. So if you just have a simple stuff of using viscozyme, have you heard of viscozyme? Viscozyme is a sort of is a commercial name for from Novozyme, that is carbohydrases, which is a mixture of cellulose and all other carbohydrates, which can break that carbohydrate and then leave the protein, and carbohydrates are broken down into sugar. So simple membrane system can remove the protein from sugar suspension to concentrate the chemicals, yeah. Or else you give just ultrasonic treatment or microwave treatment to dislodge the cell, that is to disrupt the cell, and then to use enzyme and then to separate the fractions, yeah. So there are number of different combinations. So each one itself, microalgae fresh or dried, subjected to treatment, and how much is your recovery yield is a study on its own. Fresh or dried with ultrasonics and extractability is study on its own, yeah. So all those combinations how it can be used, and lot has been published out of this, and all these ultrasonic, and this should be kilohertz, sorry, it should not be hertz. So ultrasonic bath, probe, microwave, and combination of both you will see in coming slides, yeah. So now this will give you an example how pre-treatment, this very simple experiment is which you saw the previous slide was all different combination. This was carried out on cholera vulgaris, normal system, 20 or 100% power for 10 minutes. Now again percent means nothing unless and until you know what 100% means. Yeah, intentionally we do not write all those values because just for what are exact conditions. So that means if you have 100% power is related to 130 watt per centimeter square. I will cover all these units when we go through in detail with ultrasonics. After doing these two pre-treatment, enzyme, proteases, within proteases there are multiple type of enzymes are there, that is alkalase which operates at alkaline pH, you have papain pH neutral, yeah, or you have other protease from bacterial source, bromelain, some of those enzymes they operate at acidic pH. Yeah. So, depending upon the pH you can select an enzyme, operate to see how much of protein hydrolysate or concentrates are required, and then you recover protein. If you see this is simple figure, control which was almost an 18 hours follow compared to these two, what is the protein recovery, whereas 10 minutes of this treatment compared to 18 hours of this treatment is having significantly higher recovery yield. Then further characterization into amino acid digestibility and all is not presenting it here, is not showing, just conscious of the time as well, and that is where single pre-treatment can improve extractability of it. Yeah. Another one is to do with how it looks like, the same figure. When water was used, hydrochloric acid was used or alkali was used. Yeah. Different solvent, 2 hours, 18 hours, 10 minutes, that is ultrasonics, 2 hours, 18 hours, 10 minutes, 2, 10, 10, sorry 2, 18, 10, control and ultrasonic, and you can see the differences. So, here 18 hours and 10 minutes was the same. So, that means you are reducing significant amount of time, and in order to see how it changes the structure, these were the images taken using scanning electron microscopy, which cannot be seen very clearly on this particular screen, but you can see how ultrasonics can damage the surface to improve the extractability of the biomass. Yeah. Just take the message, pre-treatment followed by this. These values will differ depending upon your biomass and intensity of treatment, and when we talk about ultrasonics we need to be seeing what is the frequency and what is ultrasonic intensity and the power. Often power is reported in publication based on the equipment power, that is completely wrong. If equipment is 1000 watt, you do not use 1000 watt, that is the power rating, that is the maximum power it will draw out. Yeah. If you use it at a lower intensity, it will be half of that. Something you have heating radiator which says 3000 watt, that means in 1 hour it will use 3000 watt per hour, that means huge amount of electricity. But if you are putting it in second regulator setting or third regulator setting, you may be using only 10% of that power. So, that power used by the equipment can be easily measured by buying a simple power meter which is plugged when you are drawing the current out of it. Yeah. So, those are the way to measure a power, and how much is delivered to the product, that depends again measurement by calorimetric method. I will cover through when we go through the cavitation part. Yeah. I hope everybody has seen ultrasonic system, whether a bath or a probe system. Yeah. So, that is one. Now, this is again to do phycobilin protein from microalgae, that is Porphyridium purpureum. This is pink color microalgae, and here this is normal shaking, and this is to do with plasma. Yeah. A specific example I loaded here only with the plasma, because you did see the plasma system, and subsequently I will be covering some of the plasma technical system here. All this UV LED light, high voltage electric discharge, atmospheric pressure plasma jet system. Yeah, the number of different ways the plasma can be imparted. You have seen one or two systems here, and wherever you go, there is everybody makes their own plasma system. It's not that complicated to make it. And the plasma can disrupt the cellular cells, like what plasma can break microalgal cells. Same way plasma can also break yeast cell or bacterial cell. Yeah. So, when it breaks the cell, it releases the compound, and compounds are present within the microalgal cell. As you can see, different powers, different gas configurations, they have different extractability of microalgal cells. Yeah. So, this is and if you have any specific interest, I can show it to you all. I can give you even electronic copies of some of those publications which you can see, and if you have any specific question, you feel free to ask. Yeah. Oops. So, now again, high voltage electric discharge either by shaking or different time points, different plasma set, that is phycobilin protein, and these phycobilin protein are pink color protein. That means PE, PC or APC, that is all phycocinins, that is color bearing protein compounds. Yeah. So, these are all different extractability yield in aqueous extract, and this was only done at the research stage to see whether it can be used or not, but obviously for commercial scale usage of plasma and all are still subject to number of different regulations, that is through novel food regulation. So, you can see on how the air or nitrogen. So, when you saw the plasma system, you can generate a plasma species by using air, nitrogen, oxygen or combination of different things. Yeah. What difference it will make? Any idea? It will differ in the reactive species. Yeah. You have seen the plasma system, what type of gas used, that will depend upon what reactive species you are making. Think if you are using only nitrogen, what reactive species it will form. When you are using CO2, what reactive species it will form. When you use oxygen, what reactive species it will form. Or if you are using air, which is combination of all these three gases, what reactive species it will form. Yeah, which is responsible for breaking of those cells. So, now once we extract those ingredients, 2 or 3 days ago one of your colleague was asking about the cytotoxicity. So, that is why whenever we extract ingredient, it is always done on cytotoxicity on cell line cultures, and to see whether those concentrations whatever is being extracted, do they have any impact on cytotoxicity, not because of ingredient, but because of the process technology which has been used for that purpose. Yeah. Does this answer the question of biological activity? That was you, right? Yeah. So, the extracted ingredient, what is their property, and if we are using any new technology, does that technology along with the ingredient causes an issue on cytotoxicity? Yeah. Okay. So, that is one which shows how we can use either jet system or gases or nitrogen. Do they have a specific cytotoxicity? If they do have cytotoxicity, what is causing cytotoxicity is A, and B, what is the concentration which will cause cytotoxicity. Yeah. So, that is all to do with cell line culture. So, this is what shows what is the cell viability and against different cancer cell lines. So, any questions so far? I just wanted to give you only 2 short examples, and then when it comes to scanning electron microscopy, again as you can see how the control looks like, and when we give a plasma treatment how cell gets damaged. Now, these SEM images are not very clear, because when the extracts are obtained or when residues are obtained, you have to dry it. When you dry it, the scanning electron microscope will not be able to pick the refined exact impact on the biomasses. For that you have to use scanning electron microscopy with clear intention or sample preparation, or what way samples are dried, because wet samples you cannot put it under scanning electron microscopy because of the moisture present. It will have different diffraction and the print will come out completely different. So, you will see much more clear pictures in next couple of slides. Yeah. Now, that was microalgae. Now, if you look at the macroalgae, whatever biorefineries were carried out, and this shows the application of different technologies, different stages of macroalgae. Seaweed remains the same, that is Alaria esculenta. Post harvest operations, what we discussed other day, washing, hot air drying or milling, soaking into 0.1 molar hydrochloric acid. Ratio here was 1:14. That answers your question, because that was the minimum what we can use to compromise between the yield and requirement. If you go 1:20, we will have a better, but we have to remove solution again. Process over here, thermal processes, that is 70 degree for 2.5 hours, hydrodynamic cavitation for 50 passes, which takes nearly about 30 to 45 minutes. First stage processing, we get a pellet and we get a supernatant. If you see in the supernatant, this supernatant was either directly freeze dried to calculate the extract yield after the treatment, ethanol precipitation to precipitate your laminarin, and the third one, membrane ultrafiltration by using 10 kilodalton membrane, because we know that more than 10 kilodalton is the molecular weight of laminarin, and after that whatever is left over less than 10 kilodalton was your mannitol and retentate was laminarin. Yeah. So, this is what was for stage 1 process. Leftward to extract alginate, 2% of sodium carbonate. Again I repeat, you can also use sodium bicarbonate. There is no standard ruling what method to be used, because that is again been optimized for that particular seaweed. Again 60 degree for 3 hours or hydrodynamic cavitation for 50 passes. Residue was protein and fiber, we left it at that. Supernatant, direct spray drying, crude alginate, ethanol precipitation, pure one, pure alginate, and then to measure the properties of alginate by using your FTIR or TGA and all those analysis to compare with commercial alginate to prove the point how is your process. Now, if you look at all these key points, that was done again with same hydrodynamic cavitator. Yeah. So, this is again tank which I already explained you. Yeah. Now, these are the extracts which are obtained directly conventional, ultrasonic bath and hydrochloric acid with alternative acid, that is citric acid, and this was a compound which was purchased from Sigma Aldrich, hydrodynamic cavitation HC and with the probe and hydrochloric acid. Diagram you saw was all related to hydrodynamic cavitation. When you go in here, laminarin, number of different treatment. When we use hydrodynamic cavitation, we had much better purity compared to direct freeze drying or ethanol precipitation. Energy consumption was very low, because when we used hydrodynamic cavitation, we were using for much lower time. When we use the heat plus 2.5 hours was having higher amount of energy, and this energy consumption is calculated based on the power drawn from the power socket. That means the specific type of power socket comes in the market, you plug it onto the power phase and then plug in your equipment. That becomes slightly complicated when you are using 3-phase current or 5-phase connection or higher amps configuration. So, that is the way you can calculate power required. Mannitol, that is sugar alcohol, membrane 40% purity, recovery rate 75%, sodium alginate 88%, recovery rate, energy consumption and its molecular weight. Protein, how much we recovered, because normal Alaria was having only 5% protein, 5 is concentrated to 17 and rest was fiber in it. Yeah. So, either we want to make a fiber rich powder, we eliminate the protein by using enzymatic processes, or use your enzymatic process to break the fibers into sugars and retain the protein, and the sugars can go for your bioenergy or ethanol production. So, this is the way how the technology can be used for making different biorefinery approaches. Yeah? And where are always the benchmark was to see what is the commercially available product. So, here it is 190 molecular weight, and there is no product sold in the market with mannitol or laminarin is available. So, here sigma, this was a molecular weight, and molecular weight what we obtained was quite similar. Yeah. Any questions so far? So, how the technology can be used? Again, if your target is only on laminarin, that is beta glucan, and how do you estimate laminarin? Either by using HPLC or megazyme kit. You might have heard about megazyme kit. Yeah? Megazyme is a kit which can help you to measure total carbohydrate, soluble fiber, or beta glucan or insoluble fiber, because in our labs we have eliminated all those analytical method which involves sulfuric acid or corrosive acid, because when we talk about sustainability and cleaner extraction, that also to modify the analytical techniques which does not include such harmful acids or chemicals. Yeah. So, that is laminarin, that is mannitol, and further usage of mannitol is to crystallize it to get your mannitol crystals, or to convert this one into maltitol, which could be a replacement of xylitol which is used in chewing gum or any of those sugar alcohols. Yeah. So, these are all different ingredients and processes used using technology. Now, this you give you another example where the wild harvest seaweed, that is Fucus vesiculosus, was used to extract fucoidin, that is fucose containing sugar or sulfated polysaccharide. Yeah. This is very simple one where you have different power levels for ultrasonic, that is 100% amplitude corresponds to 38 watt per centimeter square, that is ultrasonic intensity. How this is calculated, I will go through that process. So, here when only ultrasonic was used, these were the recovery yield. The moment we use microwave alone, recovery yield was almost 9 times. Yeah, and this is only 2 and 5 minutes, not a huge amount time. But when we combine ultrasonic with microwave, recovery yield is significantly increased. Yeah. Now, this is possible for fucoidin, but may not be possible for phenolics from microalgae or phenolics from tea, because this particular ingredient, conventional extraction process for fucoidin is boiling your seaweed extract at 80 degrees Celsius in 0.1 molar SCl for 6 hours. Yeah. Because this is not temperature sensitive compound. Yeah. That is the reason microwave, which introduces huge amount of energy and a temperature, is still you are able to achieve that particular compound. And this is one of the most expensive ingredient in terms of having biological activities as well as again antiviral activities and lot of antimicrobial properties, and plus range of different biological functions. Yeah. So, the selection of the technology here, no enzyme is used, only ultrasonic for disrupting the biomass or combination of different techniques. Now, if you look at what happens, this is not from the seaweed, this is another structural change which work was carried out on coffee silver skin, that is outer cover of coffee. Normal biomass looks something like this, flat. So, the solvent has to go in. When you use microwave, the structure is bit damaged, ultrasound damaged, ultrasound followed by microwave, yeah, subsequent, see the structure, microwave followed by ultrasound or simultaneous ultrasound and microwave. Yeah. The whole purpose here was how you disrupt the matrix so nicely so that it absorbs the solvent at much faster rate, solubilizes the solute and you remove the residue. That is the whole purpose of disrupting the cellular matrix. So, I think this is pretty clear here, and how much biomass can be disrupted. Whereas, if you have raw biomass, it takes longer to dissolve it, to expand the network, and then solvent starts going in. That is why it takes normally 16 to 18 hours to achieve the same extraction, which microwave can do it in much shorter time. Yeah. Again, the selection of technology will depend upon your choice of solvent. When you use ethanol, 100% you cannot use microwave, it will catch a fire. Yeah. When you use hexane, you cannot use microwave straight away. Polarity of the solvent, dielectric property of the solvent, all those are important features before you use any technology. Yeah. Biomass, target compound, selection of solvent, these three becomes important parameters in order to use different technologies. Yeah. So far what we talked all about was the wet chemistry and wet fractionation, which is quite commonly used for almost every processing. There is another way of using or separating proteins and different ingredients, that is your dry extraction technique. Dry, one, you have a biomass, you do a pre-treatment, grinding, milling, microwave, ultrasound, any of those treatment, and then you do the separation process. This technique is known as air classification. Yeah. At simple experiment is you take any flour or any grain, for example, grind it. Yeah. When you grind it, what will be the particle size of it? You will never measure the particle size, but if you take different staked sieves, yeah, and you know BSS sieves are there for using for filtration. So, if you take a sieve of 300 micron, 200 micron, 50 micron, 10 micron. Simple experiment what you can do, you do not need to buy a expensive air classification system. You have multiple sieves arranged on top of each other. If you put your 100 grams of either a seaweed flour, pea flour, lentil, chickpea, whatever you want to add in, if you add in here, yeah, if you shake it, what will happen? Out of 100 grams, 30 grams may be here, yeah, 20 grams here, 10 grams here, 5 grams here, yeah, the rest you can balance it out, right. So, each of the fraction, this is the particle size distribution of your flour, that is a function of your milling machine. Yeah. Now, if you do the composition analysis of this, an original flour contains suppose 30% protein, 60% carbohydrate and your 10% your ash, just take rough estimation. So, which fraction or will each of the fraction will have the similar composition profile? Yes or no? No. The reason is when you grind anything, the fiber fractions are difficult to mill it to fine powder. Yeah. So, this fraction will have larger amount of carbohydrate. Yeah. And then over here you may have more of the starch and a protein. Yeah. So, that way you can concentrate the protein, and since it will contain only fiber and a bound protein with those fibers, and if you use enzyme over here, you can recover the protein. Yeah. That could be your cocktail way of doing cocktail between dry and wet processes. What air classification does, it makes very, very fine flour, and there are different air cutters are there. So, that means starch and a protein is separated based on the density. Starch is having different density, protein is having different density. So, by that if you will see here, this is from this particular research paper, you have protein, starch and other biomass. When you pass it through, and you can see how you can separate different fraction, and then again if you have to do charged particle separation from starch, you have electrostatic separation or different biomasses. Yeah. And here you can see coarse fraction, flour and fine fraction. So, all these fine fractions, they contain large amount of protein, yeah, because they have lower molecular weight and you can grind it properly. Similar one, if you dry a fish and grind it and put through the particle size, you can see all the collagen scales and bones will be left on the top, and subsequently you will get only the protein fractions. Yeah. And then when you get a protein fraction, if you want to further purify it, then you may not need that long extraction technique or long solvent to separate the proteins. Yeah. So, the best way will be to use both dry and a wet fractionation. Yeah. Do you all agree? So, that all depends upon how you want to use different available techniques. Yeah. So, air classification separates the stuff based on density, and purification up to 50 to 60% can be achieved by only by grinding. So, you can make a protein concentrate by using dry classification. It will not be possible to make 90%, 95%, but rest of the biomass is rich in fiber. So, accordingly you can develop your biorefinery processes. That was the purpose of showing you this particular slides. Even if you are taking the P here, this over here, the coarse fraction, large amount of fiber, small amount of protein. Very, very fine fraction, you have large amount of protein and bit of fiber here, but this fiber is mainly a soluble fiber which can be grounded easily. Yeah. And if you take this particular fraction, and then here if you are using your viscozyme or carbohydrases, you can break this carbohydrate to release more of the proteins. Yeah. Or you can concentrate whatever you want. But if your purpose is to use that particular protein for making your meat analog or alternative product, it is recommended not to go for 90% protein, because you need to have water holding capacity, oil holding capacity and textural properties, and all those biomasses will provide you that type of properties. Otherwise, you get a 90% protein, then you buy a starch and to add it to make a alternative protein, right. So, protein purity should be only based on end product application. If you are looking for protein beverages for health drink, then you can be 90-95% protein, or then use a completely hydrolyzed protein which can make into the solution and you drink it as a protein rich beverage. Yeah. Purity should not be based on how pure you want to. Yeah. Like you go and buy jewelry, most of the gold jewelry is 22 carat or 24 carat. It is not ultra pure gold. If it is ultra pure gold, you can't make any of those jewelry. I was about to say abhushan. Yeah. So, based on that, level of purity depends upon what is your end product application. Any questions so far? I know I went a bit fast, so that's why I wanted to ask you a question before I move, before I continue with this. So, you have pre-treatment technologies, wet stroke dry, yeah, and previous time we studied biorefinery. Now, we are just adding technology and processes at different point to continue using the technology to improve process efficiency or to extract ingredient. So, you have seen example from microalgae, macroalgae and some examples from other grain crops. Yeah. So, principles remains the same, technology remains the same, changes in biomass will lead you or which will compel you to change your extrinsic factors like solvent, type of solvent and conditions to be used. Yeah. So, if there is no question, now I have a question, why do we need new technologies? And how we classify technologies? first and foremost condition is regulation. Yeah, it's not point number 1, is one of the point. Regulation, that means if there is a regulation that you should not use a chlorine or you should not use A, B or C chemicals, so you need an alternative. Yeah, because of regulatory aspect we need to go for alternative approaches, then you have to rely on new technologies. Second, cleaner and greener approach. In order to improve the process efficiency, to make process more greener and cleaner, technologies plays an important role. Third element is food safety and shelf life extension. Yeah. Fourth one, when we use the technology, it retains the nutrient or maintain fresh like taste. You would have seen in number of different studies, high pressure processing retains fresh like taste or maintain the sensorial properties. Other element is consumer concern. Consumers are concerned with the usage of lot of chemicals in their ingredient. So, you have to use some of the technology where you can eliminate the usage of chemical and improve consumer confidence in your product and processes. It is not important what is the product, it is also important from where it has been obtained, its geographical origin A, B, how it is being processed. Yeah, these things are also becoming critical. Yeah. Another one is there are a lot of technological advances are happening to improve the energy efficiency or process efficiency, and processors want to employ new technology to their processes, again to make more profit and to be more economical and environment friendly. It is something like when a new phone system comes in a market, you want to go for that technology. Why the previous was not working? It was working. So, new technology is coming in from 3G you go to 4G, now you go to 5G. Yeah. So, that is the one of the drivers for new technology, and one of them is environmental impact. So, environmental impact of current processes are having huge impact on environment, and you try to adopt new technologies so that your environmental impact of processes is reduced. Now, when it comes to novel food technologies, can be again classified based on the energy, that is thermal, electromagnetic. Any idea on electromagnetic? Electromagnetic spectrum is used in light based technologies, and you have your microwave or pulse electric field. Biotechnological, enzymes or microorganisms, or mechanical. Technology on its own or combination of these technologies to derive smart solutions to achieve any of those stuff. And when we use new food processing technologies, indirectly they impact all 17 sustainability developmental goals, but directly on responsible consumption and production and good health and well-being. Yeah, that means all these points, 7 points listed here, the core drivers of new technologies, how it is related to your sustainability developmental goal, and how we classify these technologies. Yeah. So, this is again the recap. So, what is the main purpose of using new technology? Reducing the waste or transforming the byproduct. This using better what we use already. Yeah. And this was mainly to unlocking the potential of seas and ocean. Yeah, or new biomasses. Yeah. So, at least after this 10 days module, at least you will learn this reduce and transform. Yeah. Okay. So, that is one, and then first one to do for reduction. When it comes to reduction, in that term, one is improving the safety profile and process efficiency. When I say reduction, reduction in usage of energy and water in the food processing, reducing the waste, or developing the new applications or new food product. Yeah, this is what meant by reduction. Now, this is a bit a mouthful of different technologies. It's good that some of the technologies are already mentioned by you guys. What are the technologies used? I am covering some of those core which were said in a first go and are well known and have lot of different applications. One is high pressure processing. What is the mechanism of high pressure processing? Very good. Pulse electric field. What is the main mechanism? And what does it mean?
We can apply electric voltage in the membrane, and in the membrane there will be a small pore be generated. It's called the electroporation.
Simple word, if you read the word itself, yeah, pore because of electric current. When given electric current, everything contains positive negative charge. Because of that it forms pores. Ultrasonics, sonoporation, the pore formation because of cavitation. Yeah. Cold plasma, UV or ozone, they have a single mechanism that is related to oxidation. Oxidation of compound which are present in bacterial membranes. Yeah. Very similar membrane principle will also be applied when it comes to extraction usage of these techniques. Yeah. And we are comparing all these technologies against heating, that is thermal processes. Yeah. That is why it is known as non-thermal processes, that is alternative to conventional thermal processing. Can you name 3 thermal processing techniques? Who will name 3 thermal processing techniques?
So pasteurization.
I told you other day it's a process. Yeah. All these techniques will do pasteurization, sterilization.
This one is microwave.
Question is, name 3 thermal processing techniques. You said microwave. Microwave is novel thermal.
So autoclaving.
Correct. Okay. You can call it. I can call it as a canning as well. Yeah. Canning is a process to achieve sterilization or pasteurization. Yeah. Canning will be a technique. I know often most of the literature and everywhere will read pasteurization and sterilization as thermal processing. No, we are achieving then what we are doing with high pressure. Yeah. We are either achieving sterilization or we are achieving pasteurization. Yeah. So 3 thermal processes. We keep giving out to thermal processing, but we have to know what are the thermal processes. Yeah. One is canning. Any other? We can, yeah. Blanching is one, correct? Yeah. Guess.
Homic heating.
Huh?
Homic heating.
Novel thermal. Yeah. That's good. Just guess it. At least will might something new might come up. Yeah. And technically canning is the one which is canning, retorting and blanching or impact heating. All these will be classified as thermal, because there is thermal, that means only you apply direct heat. Yeah. Way you apply a heat becomes a technique. Yeah. Within retorting, that is a static retort, rotational retort, yeah, agitating retort, steam infusion into the product, that all, or what you call steam infusion into the product, that all comes into thermal processing. Yeah. Whereas pasteurization and sterilization is the process which has to be achieved either by thermal techniques or by non-thermal techniques. Ohmic heating, induction heating, what else was there? IR heating, dielectric heating, microwave, all this will be considered as novel thermal techniques. Yeah, achieving the same heat effect but by using efficient methods. Now, There are two types of cell. One is vegetative cells and spores. Spores are seed of vegetative cells. When they germinate, they create vegetative cells. Now, majority of these techniques, they all inactivate cell by damaging their cell membranes. Yeah. Same thing if you consider a microalgal cell or any biological cell, non-bacteria or non-bacteria or non-yeast, you damage the membrane, that is you disrupt it to do the extraction. Yeah. Same thing applies for bacteria. Yeah. Inactivation of key enzymes modulating the growth of cells. Oxidative damage to cell membranes. Plus, these are just arbitrary values, what all is being reported in a literature, and I am not obviously a biggest microbiologist. Well, I know a bit of microbiology, but I am not an expert microbiologist. Van Impe from Belgium, Greiner, and yeah, these two are very well known microbiologists, establishing different mechanism, working on single bacteria, avoiding bacteria to bacteria combination. We came up with that one to see what are exact mechanisms for each and every technology for adoption. When we suggest any technology, we need to know what is our target. We have high amount of microbial load, we can use high pressure. That is just shooting without thinking. Yeah. If you are using a milk and high pressure is still not approved, because high pressure cannot inactivate spores. Yeah. And botulinum is caused because of spores. If you are able to inactivate spore formers, that technology can be used at commercial scale and can be approved by FSSAI, European Commission or any of those agencies. Yeah. So, thermal processing is still in use. Your normal pasteurization by using your HTST, low temperature LTLT, HTST and all those ones, yeah, or ultra high temperature. Yeah. These all techniques are still in use because some of the technology cannot achieve spores inactivation, whereas heat can kill everything. Yeah. Now, so what will be the solution? How we can use high pressure or pulse electric field or ultrasonics for liquid processing? One of your colleague is working on thermosonication. Yeah. That means combination of heat plus ultrasound. So, that means to reduce the thermal impact and taking a part of heat, and rest all is carried out by ultrasonics. Yeah. This is very similar. If I need to unscrew something on this wall, either I need such a long screwdriver made up of metal, or half I can use a wood and attach a screwdriver to it and then open it. Yeah. You can take that one as a good example or a bad example. So, you just make a use of it, or else you make a big stool and then you can use it for different purposes. Yeah. So, that is why I wrote it there, smart combinations, so that you need to know at what cost it inactivates. So, this we have already seen last time. Yeah, I am just repeating it again. Now, microbial inactivation, once you know what is inactivated, then you have to use how it is impacting your minor compounds or major compounds. Yeah. Again, similar mechanism. In most of the cases, these technologies, they have minimal impact on bioactives and nutrient. One of the reason is, even if they are degrading those compound, but these technologies are also allowing to open up the structure to release those bioactives. If suppose 10% is degradation, and we use these technologies also for extraction purposes. Yeah. That is another reason why bioactive compounds are not impacted by this. And the study, in order to see whether it causes protein oxidation, proteolysis, denaturation or oxidation or lipolysis, are achieved using these techniques, but at a condition which is not required for microbial safety. For example, for high pressure at for 10 minutes will cause lipid oxidation and protein depending upon type of the product, whereas when we use for 2 to 3 minutes, it will not cause inactivation or it will not cause the degradation. A. B. Treatment on whole product versus ingredient. If you treat juice with cold plasma, you will see degradation of vitamin C and bioactive by 30 to 40% to 50%. But if you use whole orange, you won't see that degradation. Yeah, it just depends how you are treating it. Using a hot water to wash hand is okay, but you remove your skin and then wash it, then you know what will happen. Yeah, or the ingredients versus whole food. So that's the way this, the processes are designed. Now I'll quickly go through 1 or 2 processing application and then we'll continue it in next lecture. So one is decontamination. You know what is the biggest disadvantage of new technologies is majority of these technologies are useful for liquid food product. Yeah. But what is alternative to chlorine? Because chlorine is a low cost and how you can use it. So either you have a non-thermal or thermal. Thermal, water. One of them is blanching, dipping into the hot water. Non-thermal, biological, that is your natural extract, bacteriocin or so. Physical, that is ultrasonics for washing or removing the bacteria from the surface. That is why we call it also sometimes ultrasonic bath, giving the bath to the product in ultrasonic field, it removes washing. Other one is chemicals, either ozone, plasma activated water or organic acids dipping into it. Yeah, that is contact type. Yeah, that means product comes in contact with those chemicals. Other one is non-contact, preferred will be non-contact. That means the product is not coming in contact with the chemicals or with the agent. Here is a steam. Yeah, steam bath or steaming the product. That means to inactivate microorganisms, that is non-contact. Physical, ultrasonic and cold plasma. That means same plasma in a water becomes contacted, that is plasma activated water. That means plasma, water is charged with the plasma and then to use that water for washing purposes or putting the product directly into plasma rich environment. Yeah, same thing applies for ozone. Yeah. So when it comes to seafood, if you all think about the fish or the shellfish, comes out highly perishable commodity, you wash it in the ozone water, then freeze it, wash it in a chlorine water, which is not allowed because chlorine will leave lot of residues into the product, and then to freeze it to stabilize the biomass. Yeah. So this is what to do with decontamination technologies. This is one of the example of high pressure processing. In order to prove that particular high pressure can be used for pasteurization or sterilization. Other day I mentioned about 5 log for pasteurization. Yeah, for sterilization it was 12 log, but that is only true for liquid food when it comes to non-thermal techniques. Or canning or retorting is for 12 log, which contains lot of brine solution. But when it comes to solid food, how we can achieve sterilization to achieve 12 log? There is no limit for that. Even chlorine washing gives you half log, that is 50%, or 1 log, that is 90%, or 95%, that is 1.1 or 1.2 log. So in order to prove that high pressure, this was again a system was designed using a high pressure and a temperature together. How many of you know about Bacillus amyloliqueficans or Clostridium sporogenesis? These are the bacteria whose spores, even if you heat it at 110 degrees Celsius for half an hour, it will not cause inactivation. And these are the index microorganisms used to check the efficacy of your autoclave. How many of you have validated your autoclave? Autoclaves are validated. You would have known that when you turn on autoclave to maintain a pressure, you remove all the gases, yeah, so that only steam is left, just to maintain the condition, and you check the validity or autoclave, that means calibration of autoclave by using these type of microbial film and to see how much inactivation happens. If you look at this short video, yeah, I'll remove all this, yeah.
HPT, hydrostatic pressure combined with temperature, will enhance the quality, shelf life and safety of food products, whilst reducing their global footprint. We are currently designing new equipment to introduce HPT to the global market. And this is how it will work. The food product, sealed in flexible and water-resistant packaging, is first inserted in a container and then preheated. In the next step, the preheated food is subjected to an enormously high pressure of up to 6,000 bar. This pressurization lasts just a few minutes and results in an instantaneous increase of the temperature up to 120 degrees, enough to destroy the most resistant microorganisms and enzymes. The short pressure and temperature treatment ensures that the quality of the product is hardly affected.
Now, whatever you see in this
Through
video,
extensive research,
on these
we will
two
be able to
bacteria,
determine the ideal pressure, preheating temperature and time span for each individual product,
if you
avoiding
see
pathogens
over
and
here,
obtaining the best possible food quality and safety.
time 80 to 100 minutes.
Combining our
achieve
new technology with these best practice methods,
even 2 or
both the
3
food
logs,
industry
yeah?
and the consumers will benefit from HPT
Whereas,
processing.
When
Hipster,
you use the high
making our
pressure
food safer
along with
and
such a
tastier.
temperature, we can achieve much longer log reduction at much shorter duration. Yeah. What does it mean? That means heat plus high pressure. If you go back to the literature, it will say TAPS, temperature assisted pressure sterilization, or PATS, pressure assisted temperature sterilization. That means combination of temperature and pressure. Why the limit was set as 600 megapascals? Because if we go for 700 or 800, even further we can reduce the time, but strength of the material becomes challenging. The design aspect here was whatever vessel is to be used, that is preheated to a temperature, then it goes for high pressure. Otherwise, you have to heat up the whole vessel where we are generating a pressure. By doing so, amount of energy required to heat up the whole vessel and the strength of that material, number of cycles are reduced. So, what you see here, temperature alone takes longer time. That is why you use autoclave to put some pressure and to get 15 to 20 minutes, but that time product is completely spoiled. Yeah? And this was tested on different type of soups. Yeah? That means using Bacillus amyloliqueficans. Then only we can use high pressure for industrial application. Yeah? So, this was mainly a validated study with known DSM strains which are used for testing the efficacy of sterilization, and to see the shelf life of these products. Ready to eat soups, they are only few months or 6 months when it comes to canned product, whereas when you use high pressure they have much better quality and amount of heat required is less. Yeah? So, this shows the combination of HPP with temperature. So, this had different 11 partners. One was equipment manufacturer who designed that particular system based on the conditions which were provided for soup mixes. Yeah? So, the whole crux of the story here is when we use temperature and high pressure together. The same thing on other type of sports. So, instead of having 50 to 60 minutes, we can get everything done within few minutes. What it does, high pressure will germinate the spores and heat will kill it. Yeah? When you use heat alone, then heat has to break the spore coats at very high temperature and then to kill the vegetative cell. That is why the combination of high pressure and temperature is done. If you only give high pressure to spores, their germination increases. Some of the study, if you will see, when you use ultrasonic washing of fruits, microbial growth increases, because if suppose microorganism is at one point, put in ultrasonic bath, that microorganism will detach from there and spread all over the surface. Yeah? So, that is the way what it can be used. Okay, I will quickly go through the fish example here. Very similar process applies for fish to extend shelf life. You have already seen a video what high pressure is. You have fish, you vac pack it, give a high pressure treatment, improves the shelf life. You can get up to 2 to 5 log reduction, but not long term shelf life, but short term shelf life is good. Or even if the fish is to be used for subsequent extraction, it improves the extraction efficiency, at the same time it also improves the shelf life. Another advantage is it also changes the properties of the ingredient. So, with this high pressure will stop and will continue with the application of new technologies using cold plasma in next lecture. Yeah? And remember, tomorrow morning when we start, we will start each one of you as a question and answer session. Each one have to have a question. Yeah? If you do not have a question, I will ask that person a question. Yeah? Each one will have a microphone for few seconds to a minute. Yeah? So, now you can strategically you think who should be the first one, who should be in the middle. Yeah? Okay. So, any question for today? No? Yes.
Sir, we use plasma technology for inhibit the microorganism and reactive oxygen species. But in previous slide, you saw that it also lead the DNA damage. Okay.
Yeah.
So DNA damage will lead the mutation in algae. So if mutation will be occur in algae, it will decreases the efficiency of the algae. So why we use oxidation rays and plasma technology
for microalgae? Yeah, the reason is because microalgae, when you are saying about it, now here the two things are there. When you use a plasma, suppose you have a bioreactor, you grew one species, you took the biomass away, yeah, then you have to use another species, so you have to kill those algal cells. Yeah, here plasma, when it is used for extraction, is to damage the cells, to break the cells, to release the stuff out of it. Yeah, when it comes to the damage of DNA, that is oxidation of those DNAs are one of the possible mechanism for inactivation of bacteria. That is why there was only 2+ there. Yeah, but it could not be a core mechanism of degrading the DNA. That means if you take DNA solution and give a plasma treatment, it will show you the oxidation of DNA strands. That is the way it is known as DNA damage. Yeah. It is not exactly as a mutation, but because mutation happens not only because of oxidation and from generations to generations. Yeah. So mutation is one thing, but not the genetic mutation that when you give a treatment with the plasma, DNA will get damaged, and that means next generation is going to be a different one. Yeah. Well, it does happen when it comes to microorganisms, because in food safety we study only microbial inactivation, and up to 2 log or 1 log of listeria is allowed in a food which is allowed to be consumed. Yeah. But when you see in microbiology, most of the bacteria are inactive but culturable. Yeah. And they can mutate. So that is why most of the time when we were even in COVID virus testing, we were going for PC analysis. Yeah. Because of that, it is all keep mutating. Yeah. So that is 2 different things. correct. DNA damage is not because of genetic trait. DNA damage is one of the way it does not allow it to grow. Cell rupture is there, or DNA can be picked by other stuff. So that's the two different things. We use only microalgae for extraction purposes, and in order to extract we have to break the cells. Yeah, that's what it meant there. Yeah.
Okay.
Yes.
The fish used for this high pressure processing is alive or dead?
Sorry, can you repeat it again?
The fish under high pressure processing is alive or dead?
Fish will be dead. Why you will put live fish into this? You will be in. If you put live fish, you have to go through the animal ethics. Yeah.
Yes.
Because the fish here is the fish which is harvested, packed for high pressure. This is just to show the fish. If I put a fillet, it may be animal fillet, it can be fish fillet. Yeah, this is a fish high pressured in order to improve its stability for short term. If you are going to use it for protein extraction, yeah, because if you do the protein extraction without having proper stabilization and you are retaining the biomass for certain days, it will start smelling and microbial growth. That bacteria will continue into that protein as an ingredient, then you have to process it for ingredient. Yeah. Other one is using high pressure to have fish fillet, and that fish fillet goes for pressure treatment to improve its shelf life. You will see I'll put in few more examples of fish fillet, how it improves the smoked fish and other product. Yeah.
Sir, if the water bottle there is open, we
Where is
press
water bottle?
There, in front, there water.
This one?
Yes.
Huh.
If the water bottle open, we pressure, apply pressure, the water comes out. The same way the fish we compressing, the inside stuffs may or may not come out. What happens? We any pre-heat that or directly we apply pressure. We apply any pre-heat before applying that processing, or directly apply pressure, sir.
Okay. Now, one thing is the whole fish, A, and B is the fish product that is fillet. The process involves you have a biomass, you vacuum pack it. Yeah. When you vacuum pack it, and then you go for high pressure. If you go for very high pressure, for example, if you go for 10 minutes in a fish, what you get out is a fish paste. Yeah. So, if you put a whole fish, you put it for 2 to 5 minutes, and that is the time to see if integrity of product is maintained. But it won't be the same fish as what it was before. It's something like you have a strawberry. If you put in a hot water, it won't remain the same strawberry. Spinach, you put in hot water, it won't be the same spinach. Yeah. So, there will be change in the product, and that's what is also known as high pressure for cooking the product. Yeah.
This fish example for this processing, sir, common fish.
For fish, you have a fish pate, yeah, or fish fillet. You know, when you buy fish in a market, you buy either fish fillet like salmon fillet, that is vacuum packed and then pressure treated and sold in a market. Not convinced?
Convinced. Thank you, sir.
How can you be convinced? So, I know you are thinking from the product side of it. Take any product, majority the fish in India is sold as a fish and you cook it at your home. Yeah. To make fish curry or something, you make a fish curry and you want to sell it like what you get in Haldiram and different ready to eat product. In those cases, what you saw in that video, that is cooked curry put into the packet. Instead of doing a canning process, you do it in high pressure. Yeah. Okay. So, that is one of the way high pressure is used for making direct product application. Yeah. So, we will continue tomorrow morning with the Q&A, and then we will continue with the cold plasma technologies, and then to go with the food and feed product and last day we'll cover all about soft skill IP and one or two on publication strategy and other things. Yeah.